Hello, everyone. Welcome to today's live broadcast, Developing Contamination Control Strategy. I'm Shannon Stoltz, Senior Editor of Special Projects for Pharmaceutical Technology, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by Nelson Laboratories. Nelson Labs is a global leader in microbiological and analytical chemistry testing and advisory services for the medical device and pharmaceutical industries. Nelson Labs serves over 3,500 customers across 15 facilities in the United States, Mexico, Asia, and Europe. The company has a comprehensive array of over 800 laboratory tests, supporting its customers from initial product development and sterilization validation through regulatory approval and ongoing product testing for sterility, safety, and quality assurance. So we do have a few important announcements before we begin today. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, and the slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. And I would now like to introduce today's speakers. We are pleased to be joined today by Susan Schneep and Martel Winters. Susan Schneep is the Knowledge Leader for Regulatory Compliance Associates and is Chair of the Parenteral Drug Association, or PDA, Board of Directors. Her background includes CGMPs, audit readiness, change control, investigations, supplier quality, and batch record review. Martel Winters is a Director of Science at Nelson Labs and a recognized authority and industry leader on radiation sterilization, bioburden, industrial microbiology, aseptic processing, environmental control, and tissue processing. He has over 25 years of experience on these topics and has worked with clients in all areas of the world, speaking, training, and consulting in the medical device, tissue, and pharmaceutical industries. Martel is an active committee member of many working groups within AAMI. Thank you both so much for joining us today, and please get us started.
Well, thank you for that lovely introduction. And I want to thank Martel for joining me to do this presentation. It all really started when the European Annex 1 came out, and there was a new requirement in Annex 1 for contamination control strategy. And we got to thinking about what that meant. And, you know, I first panicked, right? Because I thought, oh my God, it's another thing we have to learn and do, and we've got quality systems and pharmaceutical quality systems and complaint handling and investigations, and now we have to come up with a contamination or containment, as I call it, containment control strategy. But then when I stopped panicking and I thought about things, I said, well, we have a lot of information already. And so what I'm going to do is walk my part of the presentation will be walking you through what we already have in relationship to our process design, our facilities, our raw materials, environmental controls, the validation, container closure, and of course our quality systems. And then Martel's going to do the, what do we need? So to tie it all together so we get an effective contamination containment control strategy, he's going to tie it all together. Now, I know we started off with contamination control. On this slide, you see containment control. At one point way back in the day, in the FDA, it was actually called containment control. So I use those terms interchangeably, and if you hear me slip throughout the presentation, that's because I grew up in the era where it was containment control, but it's kind of the same thing. You're going to control the contamination from one area to another. So what do we have? Right now, currently, we have a lot of documents. We have a lot of information, right? And so when it comes to process design and microbial control, we usually have process flow diagrams. We've taken samples. We know what our environment looks like. We have established critical quality attributes, in-process controls, microbiological attributes. We have bioburden, endotoxin. We know all of this. We know what our particular particulate attributes are, defined hold times that we can keep product before we go on to the next step, whether it's aseptically filled or terminally sterilized. And we have process validations to show that we can, reproduce the product over and over and over again under the same set of conditions that are defined here. So we have a lot when it comes to process design and microbial control. With facilities and utilities, we've always got the air pressure control following the ISO system. We have water systems, water for injection, distilled water, whatever is feeding into your laboratory as well as your manufacturing site. You've got controls over that. You've already done a cleanability assessment. How easy is the equipment to clean? How easy is the facility to clean? Is there any degradation of the walls, the floors, the seams? The material flow, how do I bring it into my clean room? How do I take it out? How do the personnel come into the clean room? And what do I do with the waste? And the validation of that system. So we have a lot with facilities and utilities. When it comes to raw materials, we have the incoming specifications. We get a receipt, we get a certificate of analysis, we quarantine the material until it's been sampled and tested and approved and released by the quality unit. And we, hopefully, all of you have a supplier quality agreement for your raw materials so that you know that your supplier is reliable and is going to give you consistent product, consistent materials over and over. So we have raw materials, we have environmental controls and validation, and that's environmental controls for the areas that we're going to work so that we don't contaminate the product. So we do particulate monitoring, environmental monitoring, media fills to represent when you're aseptically filling, right? To represent that you don't contaminate the product, you're going to use media. Smoke studies, both dynamic and static, when you're in operation and when you're not in operation, so that you can see what the airflow, that you still have control and your HEPA filters are still working and the air pressure is right and the movements, and you're doing nothing in there to contaminate product. And temperature and humidity are monitored, and if anything goes wrong, typically you have a recovery plan. Personnel training and qualification. Well, any organization involved with the filling of aseptic or parenteral drugs has a gowning qualification and a requalification. You don't just qualify once, but you periodically have to requalify. And there's a disqualification if you don't if you get hits while you've been gowning, you may be disqualified for a time until you can pass a requalification. Personal hygiene is important. We all have criteria around that. Clean room practice, how you move, how fast you move. And then, of course, production microbial monitoring, monitoring your gowns, your gloves, and settling plates. All of that goes into supporting the personnel and the qualification of the personnel and how they manufacture the product. Then we have equipment validation. Of course, all the equipment we work on has to be validated, designed appropriately for its use. We have to be able to clean it so there's no cross-contamination. And we do oftentimes preventive maintenance, right? And there's always change parts available. So we know what's likely to break down, what we need to fix, how we need to prevent when things are going to degrade. We don't wait until the HEPA filters degrade before we actually go and change them. Well, it's the same usually with the glove. Those gloves are replaced periodically before you wait until the glove disintegrates and you're contaminating the product. And then the container closure system, certainly after we've manufactured the product and put it all together, we don't want anything being able to infiltrate in through the container closure system. So we do a lot of study on that, make sure there's container closure integrity. It maintains a sealed environment. We do extractable, leachable, so we know what's coming from the glass or the stopper or the bottle. Certainly sensitivity to light, moisture, heat, and cold are all involved in the container closure aspects of contamination, containment, control. And overall, we have the quality systems where we've got the trending, the metrics, the deviations, the CAPAs, everything to help us investigate when things go wrong, why it goes wrong, when we might have to reject product, under what conditions we would reject the product, how we would validate, change the system, quality control, the laboratory is under their quality agreements. Do we have any risk management? The change control for any changes? Plan, do, check, act. If we're going to make a change based on a CAPA, based on an investigation, we're going to run our plan, do, check, act model to drive for continuous improvement. So that's what we have. And what we don't have is what Martel's going to walk us through. And really what we don't have is a comprehensive plan that kind of discusses this overall. So with that, I'm going to turn it over to Martel to talk about what we need now. Martel.
Great. Thank you, Sue, for that great introduction to all of this interesting stuff regarding contamination control strategies. So the interesting thing about this, I'm going to a few key words here, and this is the idea of a holistic contamination control or containment control strategy. This is a big part of the EU Annex 1 revision, and you see there the red text here, just a few things that call out from this quote. That contamination control, so we're minimizing, of course, microbial, endotoxin, pyrogen particles, and the end of that sentence there are a series of interrelated events and measures. And then at the bottom there, that the collective effectiveness is considered together. What Sue went through is what we commonly see in most manufacturing situations, and I think the main difference is that they are not distinct things that you're considering. They're not separated out, but instead they are connected. They're interdependent or interrelated. And even though you might do the testing of some of these components separately, they do come together. And that's the focus of what we're going to talk about here. So here's a diagram to kind of give the idea. This comes from PDA Technical Report 90. And so you notice that that when you look at this big picture, the whole idea behind holistic, like I said, that they are dependent on each other, multidisciplinary, meaning you don't just have microbiologists doing the microbiology thing. You don't just have quality doing the quality thing, but that they are working together in their various disciplines to create the best possible overall strategy for contamination control. And then the tailored piece. So just because one strategy is established and works well for product X does not mean that that is the right strategy to apply to product Y. There's certainly that conceptually they'll be the same, but in actuality, how they're carried out, they may look very different depending on the needs of those products. And then deliberate. Everything that you are doing needs to have a purpose, a clearly defined purpose and documented purpose, so that that purpose is clear 5 years down the line when you're no longer involved with that specific process. That deliberate aspect is really important. So it's not just a list of things that you're doing. They're not separated from each other. And we're going to refer to this diagram here for a few slides. So let's talk about this main, from a big picture standpoint. You have the foundations down there. So as a scientist, I was very pleased to find that the foundation of all foundations was scientific knowledge. And the very bottom there, and that just makes sense, that really it is the science that must be used to drive what we're doing. But right there, hand in hand with scientific knowledge, of course, is the quality side and what they can bring in regarding risk management. And those two work together very nicely to establish not just what needs to be done, but how much of it needs to be done. For example, how frequently, or what the sample size needs to be. And then right there along with those is the personnel awareness. So this is where each person who's involved with the manufacturing process, in whatever way they're involved, be it whether it's even on the procurement side, on the transfer into the manufacturing area side, or actually doing the manufacturing themselves, that awareness drives a culture of understanding, a culture of quality. And there's a big focus there on that awareness, not just on what they're doing, but why they're doing it. We'll talk about that a little bit later as well. So the next step up there, you see you have these are the contamination controls. So what are the controls that you think are helpful as part of this overall strategy? And then in order to gain confidence in those controls, there needs to be some validation work behind those, so that you can show that this control indeed is a good representation of the contamination that you're trying to avoid in this specific aspect of the process. And then, of course, that leads to ongoing monitoring. And we're not going through every box on all of these, but that monitoring is critical. It's an ongoing thing. The science knowledge and the risk, the quality risk associated with that, those of course dictate, like I said, the frequency and the depth of that monitoring. But that ongoing monitoring and then trending of those controls is critical. And then you got this feedback loop. As you're monitoring, as time goes on, things are going to happen. And as those things happen, you'll find this is a very iterative process. So you'll find yourselves, based on the ongoing monitoring, you'll find yourselves going back, maybe looking again at contamination controls, maybe due to something that has happened over time. Maybe you need to look at one aspect of that contamination control differently. Go back to the beginning, work your way up again through either alteration or addition of controls, validation of those controls, and then add that to the monitoring process. And then you've got the big picture oversight. So these are groups and people who have to look at all this from the big picture to make sure that it's all functioning correctly and that it is meeting the intent behind the purpose for this overall strategy. So a couple summaries here at the top. These should all be working together. You might find that all those things that Sue talked about, you might find that what you're doing is actually perfect, that there might not be any changes required to what you're doing, but maybe what needs to be changed is how you're doing it or how you're using the data that you get, making sure that those fit together, that holistic mindset is in play. And like I mentioned earlier, focusing on the why versus the what. The what is very easily described in procedures, and people can read those procedures and say, well, it says to do this, so I will do that. But if they understand the why, they can ensure that they're meeting not just the letter, but the intent of what they're doing. Now, I mentioned the TR 90. That provides a roadmap. It does not repeat Annex 1. It does not replace Annex 1, but gives great guidance on establishing that strategy with this mindset of holistic and all the elements involved there. And it also helps to understand how the quality aspect, the quality risk management aspect, plays a role in development of the overall strategy. And finally there, just to help have us at least as much as possible a harmonized approach to establishing a strategy. As is normal in standards, of course, the Annex 1 speaks largely to what needs to be accomplished, not how to get there. And there are documents like TR90 that can help provide guidance on how to get there. And as an industry, if we're doing that in a fairly harmonized approach, then that's going to help everyone be better. Let's talk about some practical considerations. So what might some of these look like in a contamination control strategy with regards to some of these main components of manufacturing? So you've got the manufacturing process. So not just the question of, is it open or closed, is it single use or reusable, and is there a sterilization method involved in the manufacturing process, but in addition to having those aspects, how do they interrelate? How do they work together to result in an overall finished product that is safe and effective for the patient? And the same concept with manufacturing equipment. So you're thinking about cleaning and sterilization. You're thinking about hold times and how that might influence the finished product purity or quality. You're looking at cleanability of the manufacturing equipment. With regards to the drug substance and the drug product, so what's ideal as far as growth, or what's not ideal as far as growth? What are you implementing to ensure that either you are or are not allowing growth, depending on what your process needs to do, and what do those hold times look like to make sure that you're achieving the correct product purity and cleanliness. Microbial reduction. So if there's a requirement if there's a need to have some type of microbial reduction in the process, how well does that work? Is that meeting the intent, or is it just a process that you use somewhere else that you're throwing in here thinking it's going to make a difference? But not just that, but more focused on is it meeting the real needs of this specific product? And then you might have a product where there might be a concern with viruses, where there might need to be a removal or an activation of viruses as part of this, the drug substance and drug product. Of course, the manufacturing facility plays a significant role in this strategy. How are the products stored, and how are the products dealt with, the various materials and equipment? When they're taken into the manufacturing area, when they're removed as waste, how is that done? Is that done appropriately? The big concern I often have with this type of situation is when materials are received and before they go into storage, there might be samples taken out for assessment or for testing to make sure that it is meeting the intended need behind that raw material. And is that being done in a way that does not add contamination of some sort to that raw material? And then the movement and gowning, Sue talked about the gowning qualification and the importance of that. But how does that all sure that's kept in mind as you are establishing whatever criteria you have for that? Regarding maintenance, Sue also talked about this previously. So like I said, many of these things are not necessarily new, but the way you think about them and how you incorporate them into the overall strategy is what's different. Maybe it's the biggest difference is more of a mindset than actually what you're doing, but why you're doing it and how they relate to each other. So, and on the maintenance side, maintenance always needs to be done either preventive or unscheduled maintenance when something goes wrong. How is that done in a way to ensure that the finished product retains its cleanliness? How quickly can they return back to a situation where they can start production without there being any potential risk to the product? And then raw materials and consumables. So do you have to be concerned with adventitious agents? You have to be worried about microorganisms or viruses or other things as part of those raw materials, and what testing occurs at the supplier side versus what testing you need to do internally, and just understanding what those things need to look like. And then lastly, on the personnel side, what is their awareness from a microbiological perspective? And sometimes this is some of the harder things to have to deal with, because people tend to change over time. And as much as we would like to assume that because we train somebody once on something that it sticks with them and that they naturally grow in the understanding and the carrying out of that process, but sometimes people don't. So just a review of that awareness, or ensuring that awareness remains at the top of their minds, at the forefront of their mindset when they're in their on the manufacturing side, that they're fully aware of what they're doing, they understand why they're doing it, and they can use that information to help manufacture the best product possible. Monitoring of the controls. So this is where all these different controls that you place that we've talked about to help understand the cleanliness, if that's the right word, of whatever we're talking about, whatever product or component. How are we demonstrating that all these controls we have in place, how are we demonstrating that those are being effective? And then having appropriate levels established so that you have something to look at when you get those results back. You know what you can look at to make sure that what you're doing is functioning correctly. And then, of course, that all involves a lot of testing. And then limits. So this is the case. When I use the word limit, I'm referring to something that's ultra critical. You might have levels established where you have some flexibility depending on, for example, in the environment of certain classification of rooms. But then you have some limits that these are just things that must be met. And what are those limits, and how are those established appropriately? And am I trending those so that I fully understand how things are going in the manufacturing area? So I have a case study here. And we're not going to go through every detail here, but these are some three processes for the same product. So this is some contamination control strategies that are established for a low bioburden drug substance, and just three different ways that you might look at that manufacturing process. So we'll go through these one at a time. You notice the diagram here on the right, and something that the reason we have these colors have been added. You see the red colors and the green colors in that diagram, and the idea behind that, not that red is any different than green, but the fact that they're colored means that because of the way the process is established, those are things you're going to have to pay special attention to. The things that are in white are things that almost by default they'll be addressed. So with process one, you see this is a more simple process. So there are open steps at the beginning, at the end, for example. And even though, so you do have a great environment, good personnel, you have good controls, and again, we're not going to go through all of these, but some of these aspects here, like you're using reasonable equipment with complex pathways, meaning we might be using the more traditional approach, which works just fine, but the more traditional approach of the stainless steel tanks and then the piping around the room. And those work great, but the reasonable equipment does carry with it some potential issues that need to be addressed. So because of the way this process is established here on the left, by default, there will be more requirements on the monitoring side of things to ensure that the contamination controls you've put in place are resulting in a safe product. Now, as we scale up in approach, so process 2, this is one, maybe instead of having open steps at the beginning, at the end, there are fewer open steps. There are only some open steps at the beginning. So after the beginning, the rest is a closed process. So you have some additional controls there. And so there are other aspects here where maybe you've increased your partnering with your vendors to better understand, to better define and understand what data they provide you when you receive products. You've got inspection of the consumables, and for example, as you're using the bags, you're pre-inflating those bags, doing a leak test as part of that process. So we've added some steps here, taken some of the steps in process 1, made them a little bit better to enhance the overall strategy of contamination control. And then we have process 3, and you see in this case most of those boxes are white on the right hand side, and the concept there, like I said, is we've engineered the process with a contamination control strategy in mind, such that many of these aspects of contamination controls and validation and monitoring, many of these aspects are taken care of simply by the way you set up the process. Now, this might be a more complex process as far as how it's put together, but it results in an easier way to assess those products. For example, you have no open steps. It's totally a closed system. And you've done even a better job partnering with vendors, for example, to have really good quality inspection in place, both on their end and at your end. You've, at the very bottom there, you've added treatments. So you've added some level of sterilization, whether that's enhanced sterilization of the individual components or maybe even a terminal sterilization process. And the concept here is to get the idea across that as you better engineer your contamination control strategy, you realize that more aspects of the overall, in this diagram here, more of those aspects of this overall process become easier to address. And you might have fewer things that you have to pay very careful attention to in order to result in a safe finished product. So takeaway messages here. First of all of these practices or controls or steps that Sue talked about at the beginning, that one of the main shifts as we're talking about the Annex 1 and the holistic approach is that they are now interdependent. They work together rather than being separate ideas. And they're everything you do is very deliberate in how it interacts with each other and how each thing helps to result in a finished, safe product. And again, that adding this end-to-end approach to this discussion. Anytime you're talking about a holistic contamination control strategy, it does not start and end in the manufacturing area. These strategies start at the very beginning, even selection of the materials. If you can select materials that help, that can lend themselves to both materials and designs that lend themselves to terminal sterilization, you've taken a huge step towards reducing risk in the finished product. So thinking end to end as you're going through this is a big part of this. And then formal and documented. Again, this idea that it's not just something that you do, but the reason that you do it is well described, and how they work together is well described, so that a reviewer can look at that and understand it well, so that those involved in the manufacturing side, the personnel, are all aware, they understand the why behind what we're doing. Okay, those are the takeaway messages. Sue, I'll let you wrap us up.
Thanks, Martel. Could you advance the slide? I seem to, thank you. I wanted to thank the members of the PDA Technical Report 90. These are the people who put their expertise together and formulated the report. We've just taken and allowed us to use several of their slides. And we've just taken it and tried to explain to you what the requirements are in terms of Annex 1. Next slide. And I always like to end with a final thought, and my final thought is always, quality is always fashionable, and that was said by Boy George, believe it or not. I think he was secretly rooting on our industry. So remember, quality is always fashionable, and every time we get one of these initiatives, we need to take it, embrace it, and make sure that we do a quality job with it. I want to thank Farm Tech and Martel for participating, and we are ready for questions and answers.
That was wonderful. Thank you both so much for that informative presentation. So before we get started on the question and answer session, I would like to quickly remind our audience how they can submit questions. You can submit questions by typing them in the Q&A box, which can be found directly below the video player. So our first question from the audience is, how is CCS different from safety testing methods implemented in the process?
Well, I don't know, Martelli, if you want to start this off or I'll start it off, but it's different. You can implement safety testing, you can implement parameters like pH testing, those kinds of things during your process. This is taking a holistic view. So it's tying all of those safety testing methods, the gowning, the safety methods that you take when you're gowning, those precautions you take there, and it's putting them in one overall document. So that's how it's different. It's not focusing on one safety test. It's not focusing on one activity. It's taking the holistic viewpoint.
Yeah, and maybe just to add to that, I think kind of what Sue mentioned there and what I mentioned earlier in the presentation is that it might not be so much what you're doing, but why you're doing it and how you're looking at the results. So a safety test, I might be doing a safety test hoping it will tell me that the process is functioning properly. Whereas when you have a holistic approach established, then the safety test is not something that you're saying, I hope the data, I hope these results come back good so I can release product, but you're saying, you know what, this is a well-defined, well-established, well-characterized process, and these results are just going to be icing on the cake. The cake is established, we're in good shape.
Yep.
Perfect, thank you so much. So on to our next question. Is it an obligation to implement CCS in a DS manufacturing plant, not sterile manufacturing, having C and D grade areas?
I don't know that I'd call it an obligation as more as good business sense. Obviously, when you look at Annex 1, they're talking about sterile medicinal products. If you're making a drug substance, right, and it's not reputed to be used in parenteral applications, you probably don't need as much revolving around your contamination control strategy, but you should probably still have one because you are going to get testing. There is going to be testing done on your product when it is brought into a facility to make sure that it's clean and doesn't have any dirt or germs or bugs in there. So you're probably, I wouldn't say it's an obligation. I would say it's a good business sense. Martel?
Yeah, I just add I would want the main driver to be, what do you anticipate your customers may be needing? And how high a level of contamination control do you think any of your customers might need and then addressing your contamination control strategy based on what you might anticipate that needing to be.
And I think it's especially important. I'm assuming that the drug substance line, you're going to make multiple drug substances on the line, that it's not a dedicated line. In that case, part of your containment control strategy is certainly going to be cross-contamination of one active ingredient to another when you switch lines, right? So you have elements of it already there. Why not put the foundation around it, right? Why not put the whole thought process behind why you're doing the different cleanings that you're doing?
Thank you. So what are suitable CCS for the removal of adventitious agents, especially prions?
Sue, should I take an initial shot at this?
I'll let you take an initial shot at that, yeah.
Yeah, prions are interesting. They're very resistant to most forms of sterilization, except for generally high heat or fairly caustic chemicals, both of which tend to not be good to a drug substance or drug product. Generally speaking, the way that I would address this is the same way that they address prions and other adventitious agents in the tissue industry. In that industry, that they don't test for prions. They're very difficult to test for. There are no good test kits for all forms of prions. So what you do is you ensure that controls are in place, that the source of the materials you're getting exclude the addition of prions through all the raw materials, just ensuring that the raw materials are correctly sourced to just make it so it's not an option to get that in. That tends to be the best approach. Sue, would you add anything to that?
I totally agree with that. It's exactly where I would head. It behooves you to take a look at your suppliers and what they are doing, and to ensure, and that should be part of your rationale when you establish your strategy, right? Your CCS. I've looked at my suppliers with regard to prion. If you have that particular concern, right? Look at your suppliers and do that upfront work to guarantee that they're taking care of things on their end, right? It's kind of like supplier qualification.
Exactly.
Perfect. So another great question that came in, can you share an example of how a holistic contamination control strategy would look at things differently than the traditional approach?
So I share some thoughts on that.
Start us off and we'll just kind of jump in the conversation.
Yeah, so largely what we're looking at there, the holistic approach means a couple of things. One is that you've got the end in mind, and the end is helping to drive the process. And then the process also that they interact well. So for example, let's say from a microbiological perspective, contamination control, the question is, what is the required level of microbiological control in this finished product? Does it need to be sterile? What is the sterility assurance level? Does it need to be clean but not sterile? And with that endpoint in mind, then what level of control is required for each of the raw materials or for the overall process or for the manufacturing areas? And of the many variables you have that can influence contamination control, which of these many variables require monitoring? And what controls are appropriate for each of these variables? And then how do these variables interact? So for example, if I have one raw material or one process that might pose a greater risk for contamination control, despite my best efforts to control it, if there just happens to be a greater risk because of the raw material or because the process might have to be manual, then that will influence how much risk I'm willing to accept for other processes or other raw materials in that process. And that way, and also what other monitors I might want to be, how much monitoring I might feel I need to do on these other aspects, so that I can further mitigate risk in some areas to help compensate for the potential risk of one specific process or raw material. Sue, anything to add to that?
No, I think about that. Taking a look at that blending, you might not have done traditionally. You might have just received the materials, but not looked at how they interacted and what bioburden level each one had to be able to control that to the end in mind of a clean, sterile product. I think that's perfect, Martel.
Perfect, thank you. So then we can move on to the next question. Do my suppliers need a contamination control strategy?
Probably, the answer is probably not necessarily. Depends on the supplier. The wishy-washy answer is maybe, maybe not. If the supplier's making materials to go into a parenteral product, I would think it would be, again, good business sense for them to have a containment or contamination control strategy. It's just, let's put it this way, whether you're the supplier, the manufacturer, it makes sense to have a contamination control strategy. It's going to help you when you have an investigation. Suppliers have investigations into failures. It's going to help you when you have a CAPA. So while it may seem like an additional burden, I think it goes to that concept that Martel and I have been discussing. We have a lot of stuff. Now let's put the structure around it so that we have all these pieces and parts talking to each other, and we have a much better handle on how part A, the equipment affects the manufacturing, affects the raw materials. And that's what the CCS is intending to have us do, right? Look at everything together rather than individual pieces.
And I just add that they're based on your understanding of risk in the process and which raw materials of the process have the greatest potential influence on your level of control, then the ones with the potential greatest risk, certainly you should be talking to them about having a good strategy, because otherwise, when you're testing these raw materials as they're coming in, you're going to have stricter controls on what they might be used to providing, because for you, it's a greater risk aspect of your overall process. And if they're not aware of that, then you're going to be rejecting a lot of batches of product, so that you'll be prioritizing that based on that risk as well.
Agreed.
Perfect, thank you. So does a CDMO need to prepare a contamination control strategy for each client?
No. Okay, that's the short answer is no. I wouldn't even advise that. CDMOs are in a very specific niche in that they have multiple clients, right? But when they formulate their contamination control strategy, it should be with all clients in mind. And if a client has a specific request, and this does happen in the CDMO world, right? I have this specific requirement. It's not good to step out of your processes and make a different document to accommodate someone else's specific situation. You want to have a document that stands up for all your product and all your clients. Otherwise, which one do you pull out? It's kind of like if you have multiple containment control strategies, one for each client, which one do you pull out when you're audited for your general GMP? It's very complicated if you start breaking off things and doing specific requests for specific clients. The more you can drive it to one containment control strategy fits all, the better off you're going to be from a regulatory GMP perspective. Martel, add to that. You deal with a lot of clients.
Yeah. Working at a contract lab, I am very aware that the more differences you have in how things are done, the more likelihood is that you're more frequently going to not do one of them. You're going to deviate from this special established process. And sometimes those are necessary, but just like Sue said, in our lab, we want to implement
the types of processes that we can demonstrate that our testing is being done correctly. We want to do that as consistently as possible across the board to avoid deviating from special procedures.
Great, thank you. So our next question coming in is really interesting. It's a two-parter. We have established a contamination control strategy and performed a risk assessment. How do we best ensure the actions identified from the assessment are taken? And then part two, could we prioritize them and should we justify ones we can't or don't take?
Yeah, I'll take a first shot, Sue, because I read through this 4 or 5 times and thought about it. So you have a CCS, great. You perform risk assessment, great. So what you're seeing here is exactly what we see all the time. Risk assessments identify tons of potential actions to take. And now the benefit to a well-done risk assessment is not only do you end up with this huge list of potential actions, but they also are naturally prioritized by the system as far as risk goes. So now you've got those prioritized as far as risk. So the question you might be asking is, well, we've got these 7 or 8 items, for example, that have the highest risk priority numbers or that end up with the highest values, however you're doing your assessment. And so I've got these 7 here that, according to this process, say that we really should address them. And what if some of them, how do we prioritize those? And what if some are just too difficult? And that's a very valid question. And ideally, you're able to address all of the higher risk items that come out of your risk assessment, and you're able to address them through either controls or monitoring or both. And but if there are some that just due to the way the process works or the type of raw material, there might be some things that you just have no control over, or you have little control over, and you can't implement anything to gain more control. And that's part of life sometimes. And kind of the example I shared earlier, that's where that comes into play, where that means that you just understand, okay, this aspect is going to have greater risk. Well, we need to tighten down the risk in other areas so that the overall risk is still manageable and acceptable. Sue, anything to add to that?
I think the only thing that I would add to that is the question on justifying the ones that you don't take. Absolutely. Justify why you're not taking it, explain why you're not mitigating that risk, and don't lose sight of it, because you may have an investigation that may actually be one of those risks you didn't take coming to life. You don't want to lose them. Right? You want to have them somewhere in your system so that in case you have an investigation and go, oh, wow, that's the one we didn't mitigate. You've got a record of it. The only other thing, the piece, how do you ensure the actions that identified from the assessment are taken? You need to use your quality management systems effectively to make sure that you're implementing the changes that you need to make from the actions that you identified in your risk assessment. So you've got to tie all of that together. And if you have a robust quality management system, you should easily be able to identify that you've taken action and you have embedded or you have a better contamination control strategy. Those would be the 2 thoughts I would add.
Perfect. Thank you both so much for that answer. And how can quality culture be addressed and evaluated as part of the foundation of the CCS?
Well,
you're not supposed to laugh, Sue. That's a good question.
Quality culture, to me, that's kind of the overall of everything. If you have a quality culture that is open and transparent, right, you're going to have CCS nailed down and people are going to be receptive to it. And so quality culture kind of overrides everything for me, right? And the quality culture has to be open, honest, People have to be able to speak up when they notice stuff, and the organization has to take action when they hear of potential issues. And that would be, that would be what I would say about that. Martel?
Well, and maybe just to add, how is it evaluated, and that can so evaluating a quality culture, it's not something that you can do with simple metrics. Sometimes that's more an issue of understanding certain metrics can help you understand that. For example, the quality culture should drive certain quality aspects of the finished product. So if the finished product is turning out perfectly or very few issues, then that's likely being driven by a successful quality culture. And if you're seeing frequent issues with one aspect, there might be something you can use to supplement that quality aspect to improve the finished product. That should be the main driver, because it's all about the patient, and the finished product then can help to drive that finished product.
Absolutely.
Perfect. Thank you. So on to our next question. Is the FDA requiring CCS according to EU Annex 1?
Oh, that's very interesting. FDA is part of PICS, right? The Pharmaceutical Inspectorate Scheme, right? Cooperative. And so they acknowledge CCS. And they used to call it, and if you noticed in the beginning slides, I kind of flip-flop between contamination control and containment control. And a lot of the elements that we have put in place for CCS, which are, came under containment control. So I think the FDA would be looking for that type of document. They might not call it CCS, they might call it CCS, but they are going to look for that kind of document that ties all of the elements of your contamination control together. And just like they're looking for, robust quality management systems that do plan, do, check, act, that are always doing, continual improvement in your organization. I think they would consider a CCS to be part of that continuous improvement operation.
Yep.
Did you want to add anything, Martel?
No, I don't. So based on time, should we take one more question, do you think?
Yep, we can squeeze in one more. So as for the final question, once I establish my contamination control strategy, how often do I need to revise it?
That's kind of a loaded question, right? So I want to say as often as you need to. However, if you find you're revising it every month, you didn't do a very good job on your CCS. But if you're only doing it every 2 years, maybe you didn't do a very good job on your CCS. So I think you need to put it on if it doesn't come up for normal revision during normal investigations and procedures, you may want to put it on like an SOP schedule every 2 years, whether I've made a change or not. But if you find you're revising it too much, I would go back and redo that CCS, because if you have to revise it all the time, it probably wasn't very well done in the first place, right?
Yeah. You hit on the main word that came to my mind was change. That should be an obvious driver for revisiting that. Yeah.
Perfect. Well, thank you both again so much for that lovely presentation and for taking the time to answer all of these audience questions. With that, it looks like it's already time to wrap up, so I'd like to thank the audience for attending and participating in today's event. And I would also like to thank our sponsor, Nelson Labs, for making today's educational webcast possible. We'd like to ask everyone in the audience to participate in a brief survey that will appear on your screen after today's presentation has ended, and you'll also receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see you all next time. Thank you and goodbye.